Hello and welcome to today's Galaxy introduction. My name is Teresa Müller. I'm working for the Freiburg Galaxy team, and today I want to do the Galaxy introduction from peaks to deans with you. Currently, I am on the Galaxy project training material website, and here you see under Start Here the topic Introduction to Galaxy Analysis. And if you click on this section, it will open up all kinds of Galaxy introduction videos. And today I want to do the From Peaks to Deans tutorial with you. So select this video by clicking on it. Sorry, not the video, but the tutorial. And the tutorial opened up. So as I said, today we're going to learn about Galaxy. But to make it more fun, we have a scientific question within that video, which is to get from peak regions a list of gene names. But as I already said, the main objective is to familiarize yourself with the basics of galaxies. So how to obtain data from an external source, how to run tools, how the history works, how you can create workflows in Galaxy, and how to share your data. But it's always more fun if there's also like a real life scenario. So therefore, we picked one paper that was investigating the histone acetyl transferase MOF. This is a protein which is regulating embryonic stem cells. And within this paper, they wanted to investigate where this protein MOF is binding on the DNA. And for this, you can use the ChIP-seq protocol, which is a high throughput protocol detecting binding sites of proteins and DNA. Within the paper, you have given a location on the GEO database, and there you get a result file that looks like the one you can see below. But this is not really meaningful because we don't have any gene names. And this is what we want to find out in this tutorial, how we get from this just numbers to actual gene names. But I will guide you now through this tutorial step by step. As this is a Galaxy introduction, let's jump to your favorite Galaxy instance. I will use galaxy.eu, but please feel free to use another Galaxy instance. You can do this analysis without logging in, but you will lose all of the history and every step you did if you are not logged in and you don't have an account. So to avoid this, please either log in or register if you did not already do on this Galaxy instance you want to use. So by clicking on login or register, the page that I'm currently in will open up. And if you have an account, you can just paste in your account and log in. So this will now open my account. You can see I'm now logged in at the top right corner. So here you can already see we have the Galaxy interface, and we have three main panels in this interface. So on the right side, we have the Galaxy history, where at the moment my history is empty. You might have another history open, could be, or maybe also you see an unnamed empty history like me. This history will collect all of the tools you were running, so every step of your analysis will be tracked within this history. Then on the left side, you have the tool or the interaction bar, which, for example, hosts all of the tools. So if you click on tools, you see the different chapters where all of our tools are sorted into. So, for example, if you want to do text manipulation, you could open this tab and then you see there is a tool which is called Add Column. And if this is something you want to do, you can select the tool. And in the middle panel, it will open up the interface of this tool and you can select whatever parameters you need. So throughout this tutorial, you will get to know some of the Galaxy tools we need for the analysis, and I will guide you through this step by step what you need to enter here. Then, for example, we also have the interactive tools in the activity panel, or where you can also click on the upload to upload data. You see your visualizations here, your workflows, the libraries which are available on the Galaxy instance, and many more options. So I already said that, or you already have seen that if I click on a tool in the middle panel, the tool interface will open up, and here you can now select all of the options and the parameters. So the middle panel is your main panel where you will do all of the interactions and work you are actually doing on Galaxy. Okay. There is another very nice trick. So if you are within the Galaxy instance, you can also open the Galaxy Training Network homepage by clicking on this training or this head icon. And this will open up the interface. And if you are already within a tutorial like I am, you can click on the home button, so the Galaxy Training exclamation mark, to get to the front page. And now again, we can select the Introduction to Galaxy Analysis topic and search for the Peaks to Genes tutorial and click on this. And this will now, within the Galaxy instance, open up the tutorial. So you can see we already got to know the interface. So our history panel, the activity bar, and the central panel. And now we want to check how to create a history within the tutorial. So if you want to jump back to the to Galaxy, just click outside the GTN page and you will be transformed back. If you don't have this empty history like me and want to open a new history, you can just click on this Create History button once, and then it should open up an empty history like this one. If you, or no, not if, you should always first name your history with a meaningful name by clicking on the pencil. And then, for example, type GTN tutorial from peaks to genes. You can also add some annotations if you want to. The name should be a meaningful name that helps you to find the history later on and to know what you did within the history. To save the name, click on the Save button below. And voila, we have a name for our history. So let's start with the tutorial. First, I will open it up again, and the first step is to upload data. And we want to find this file on the Geo database. For this, please open up the Geo database by right-clicking on your mouse and just open this link in a new tab. So this will open the geodatabase in a new tab. And now we can find the file name with the accession number and then mov3 out hp txt.gz file. And there are several options on how to upload your data now. You could download it locally to your computer and upload it here. We also have it already within the GTN library stored in Galaxy. But I will show you a third option today on how to import this data now into Galaxy, which is using a link. So again, if you click on the HTTP with the right side of your mouse and then just copy the link, you now have the link copied, and we can go back to the Galaxy instance and click on Upload Data and go to Paste and Fetch Data. So we clicked on Upload Data and then on Paste and Fetch Data. And here you can just enter the copied link. And we can also already say that we want to have the file type, which should be interval, used for the upload. You can also give it a different file name, but I will leave the new file name for now. I can show you how to rename it later. Okay, with this we have the link and we did specify the data type. And now I can press start to upload the data and close the upload window. You see at the moment this job is gray because it did not start uploading. Once it starts uploading, like now, the job will turn orange or yellow. And once it's uploaded, this box will turn green. And then we can also have a look into the file. So I will pause for a little bit until the upload is finished. Welcome back. So my file turned green and therefore the data is uploaded. And what we can do now is to rename this file, because at the moment we only have the link, which does not look nice. So let's name it to the file name that is given in the tutorial, which is this accession number mov3 out hp txt gz. I will just copy paste this one. So first copy, of course, from the tutorial, and then I click on this pencil icon, which opens up the Edit Attributes tab in the middle panel for me. And here you see the name of the file, and this one I just delete and paste the file name I want to give and scroll down and click Save to rename it. And when I expand the green box of the file, I see more information about our file. So we already identified the file format as interval, and there is no database assigned at the moment. So it might be also nice to already assign a database here, because if you further want to process this file, many of the tools need to specify database. And we also will need it in this tutorial. So to do this, you can go again to the edit, either on the pencil or on this edit button. And So I click on it again if you jumped out of it. And then you will see one section that is called database build. And here, if I open this one and search for MM9, which is one of, which is stands for mouse, I can select this one and again click on save. So by specifying the database now to MM9, which is the same one as it is specified in the paper, we can redo the analysis as it was done, hopefully, in the paper. Okay, let's have a look into our data a little bit. So by clicking on this I view, we can open up the file in the middle panel to get a preview of the file. So you see the first column has 11111. If I scroll further down a lot, it will continue with 222 and so on. The next two columns, they look for me a bit like coordinates. And therefore, we also choose this interval format, which is a galaxy format for representing genomic intervals. This first three columns in this format are fixed. So we have in the first column the chromosome, and the next two are the positions. And you can have as many follow-up columns as you want. Therefore, this file format makes sense for the data set we have at hand now. Because this one, as it is already specified in the name, is giving us our peak regions. So the regions where we have amplified signs of reads that we detected via the high throughput protocol. And to specify that this is our peaks file, we can also give this file name a tag, because when you specify a tag now, this will be propagated throughout your whole analysis. So every time this file will be used or worked on, it will also get the tag peaks. So for this, click on this add tag line, and we can just specify hashtag peaks and press enter to give it the tag peaks. So with this, we now already have our first file within the history, and also specified the database, the format, and a hashtag. So having now our peaks file is great, but as I said, we would like to also find out what genes are behind these regions. So what gene is falling, for example, into this first region area. And for this, we will need somehow a file that tells us where is, so on what location our genes lie on the genome. So we need a genome file. And for this, for example, we can use the UCSC tool to get this information file. So if you go to your tools, for me it was already open, and then in the search panel type UCSC main, it will select in the suggestions the tool called UCSC main table browser. And this file you can open up now, and it will automatically redirect you to the genome browser of the UCSC website. I can verify that I'm a human and hope that I get redirected, which is happening now. And now we can select what kind of file we want to have. So we know that, yes, we have a mammal, and we know that our data comes from mouse. So we don't want human, but mouse data. And we also know that the assembly was MM9, not MM10, as specified here. And as group, the gene and gene predictions are fine, but we want to have as track not the UCSC genes, but the RefSeq genes. And also from the table of ref genes, which was automatically specified. Then the regions should be genome, which is fine. So we want to have the whole genome. We want to have the output format in BED format. This should be sent automatically to Galaxy, and plain text is fine. So with all of these settings, we are good to go and can create our file. So I can click on Get Output. And I'm redirected to the output options. Here you need to specify that you want to have the whole gene and not a specific region. But this is already selected, therefore I can send the query directly to Galaxy. And now I'm directed back to the Galaxy instance and hopefully get the file in my history. You see that I have a gray file here, which is uploading now. So I will pause the video again for a little bit and come back once it's uploaded. Welcome back. My file is now uploaded and we can have a look and expand the information of the file. So we have the output from the UCSC main tool, which is also the title of this file. And the format is BED format and database is MM9 as our peaks file. The name of this file is not that meaningful, and therefore we can rename the file to give it a more meaningful name. So for this, I again click on the pencil icon. And as we know that this is our gene file, so we get the gene information here, let's just call it genes. And I can click on save. And what I can also do is again give it a tag. So I press on the add tag line and put a hash and then genes and press enter. So now this genes again also will be propagated every time we are working on the gene as an input data. Let's have a look at this file as well. So we know that is the BED format, and the BED format is a bit more restricted on what column entails what information. So the first one, as in the interval file, holds the chromosome, then the start and the end position. Then we have a name, a score, the strand, Then we have tick information, so where exons could be starting. We have how many there are, and so on. So this BED file is more restrictive. But of course, as Galaxy know that this is a BED file, it can already give you a header for this line with meaningful names, which it could not do for the interval file, because here it's not restricted, so it cannot be automatically assigned. It would be also nice to compare these two files, and for this we can use the side-by-side view. So I can enable this tile icon and again click on the eye icon of the two files, which opens up two files, and now I can open them side by side so we can compare the content of the files. So you see the first column both holds the chromosome, but we have for the genes file CHR1, and in the interval file we only have one. Then the next lines are the positions, which seems to be similar. However, we don't know how this file is treating the sex chromosomes. So we already know this distinction that the CHR is missing in the interval file, but we know from UCSC it will specify the sex chromosomes as X and Y, but we don't know how it looks for this output file. So it might be nice to check this to make the files fully comparable, because what we want to do in the end, we want to overlap those two files and see where we have overlaps to get for the regions in the interval files the names of the genes. Okay, so I will close those two files and also again uncheck the tile view and open up the tutorial, because the tutorial will give us a suggestion on how we can check the last lines of a file to see whether the sex chromosomes are called X and Y. So we are on the hands-on box view end of file, and this hands-on box suggests us that there is a tool, select last line from a data set. And I opened up the tutorial this time because we have a very nice feature here now that if you hover over this box, you see that it turns blue, and you can just click on it, and by clicking on it, the tool will automatically open with exactly the same version which was also used in the tutorial. If you want to compare and get exactly the same results as within the tutorial, it's always good to use, or you should use the same version that was used in the tutorial to make sure that you are not getting different outputs. So we want to specify now as a first parameter the input file. And this one is not our genes file, but the peaks file. And we want to keep the last lines, which is fine, but we want to keep the last 100 lines. And let's run the tool and see what the last 100 lines give us as an output. So my run is finished and I get an output. of the last 100 lines. So you see here that we have the last 100 lines only selected, and I can click on the view icon again. And you can already see by this little preview here, but now it's also in the middle panel, that we have chromosome 20 and 21 specified as sex chromosomes and not X and Y. And to now interject both of these files, as I already told you, we need to have those lines very similar, and therefore we need to rename the chromosome line to CHR, and then chromosome 1, chromosome 2, and so on, and also replace the 20 and 21 by X and Y. And we can do this by a tool which is called Replace Text. So I open up the tutorial again to go to the tool that is suggested in the next hands-on box. And I click on it. And we know that within our peak file, now it's important to select the peak file and not this 100 that we selected last. So again, select the peak files, and we want to replace in column 1 the pattern of, so now we need to find the numbers that we have, so 1, 2, 3, and so on. They are changing, and therefore we use a regular expression. So we want to find a pattern that is within the alphabet from 0 to 9, so whatever number. And we can also have more than one number. So this regular expression tells you in the parentheses, in the brackets, whatever alphabet it should search for, so this time numbers, and it can be more than only one number. This is specified by the plus. And we want to replace the pattern that is found with CHR and whatever number was found, and this we can specify by just setting an end sign. And there is no space in between the CHR and the number. Okay, and with this, you already specified that you want to add a CHR to the chromosome line. And we can run this tool to see if this really works. And the CHR will be added to this line. The tool did finish for me, and you see that we have now the CHR1, so the CHR added in front of the 1 here, and also if you scroll further down, you will see it that it's for all of the chromosomes we have a CHR in the beginning now. But we already know that we also need to change the sex chromosomes. For this, we can rerun this tool. And to rerun a tool, you have this turned arrow to rerun again. So just click on this button, and it will reopen the tool run with exactly the same parameters. So we entered here our peaks file, and we changed in column 1 the pattern. This time, as we already have changed the CHRs, we select our last file, so the 4th file in our history. And we want to change again in column 1, but this time we want to find all lines that have CHR 20 and replace this ones with CHRX. And again in column 1. And to not rerun the tool, you can also insert more replacements. Options already here. So again, we want to change in column 1 the CHR21 with CHRY. And now we specified to change the sex chromosomes, again, both in column 1. And if you did it exactly like this, you can run the tool and hopefully now get the correct output or the correct first line. You need to compare your data, so your peak data, with the genes file where we have the CHR. So my run is finished now. And at the moment, I see again the same output file, which is fine. But we want to see whether our changes really did work. And to do this, we can rerun the select last lines. So I just go down into the history and select the rerun job again button. And this time I enter our last output and keep the last 100 lines. That is fine. I just rerun the tool to see whether I get the same output. In the time that this tool is running, we can already have a look if we have used the correct file. So as we did not change anything of the content, we only added the CHR and exchanged some numbers. We should have exactly the same amount of lines. So this 4,800 something, which is the number of our last output file. And if I scroll down and see what number we have for the peaks file, I can be relieved because we have exactly the same number. So I did use the correct file, hopefully, but it looks like I did. And let's check that the chromosome changed also with our select last call. And you can see that we now have chromosome X and chromosome Y. So this is very good. We changed our peaks file to the format that we want to have it. And because the name of this tool run is again not so meaningful, like replace text on data 4. What is data 4? We don't know. Therefore, we will give this file also again a meaningful name by clicking on the Add Attributes pencil and rename the file name to peak region. And I click Save to change the file name. So I have still the tag, which was propagated, but now I also changed the file name to just, then it will be easier later on to find the correct file. Now we want to do an additional step, and for this I go back to the tutorial. So we did the replace text, and we now have the two files which we could intersect, but as we know, this morph protein could be a transcription factor, meaning that it binds to promoter regions, we can add a little bit of upstream of each of the genes, so a little region upstream of each of the genes, to also catch areas of these peak regions where the protein bound to those promoter regions. At the moment, we will not get these areas as this upstream region is not included in the genes file. So to make this a bit more clear, we have a little depiction in the tutorial. That's why I opened it for you. So there is a tool which is called Get Flanks. And this tool is functioning like this, that you have an input region. So the input region in our case will be our genes. And you specify how much of this input region you want to have by an offset. And then by specifying the length, you also add the length of the upstream region you want to add to your output region in the end. So to do this, we open up the tool Get Flanks. And here we can specify now that we want to have as an input our gene file. So not the peaks file, but the gene file, because now we want to add to the gene file our a region upstream that hopefully covers the area where the promoter region. And we want to specify the region around the start site as it was depicted in the tutorial. So we want to start from here and then specify an offset and then a total length that includes the amount of offset plus the additional region we want to add on top. So we specified the offset with 10,000 and then the full length of the flanking region with 12,000 because we are adding 2,000 nucleotides upstream of the gene, because in this 2,000 area region there should be somewhere the promoter region located. Okay, so we specified our gene file. We start from the start of the gene. We specify upstream the location region and an offset of 10,000, and the total length is 12,000. And if you specified all of this, you can run the tool. So unfortunately, my job somehow got stuck, so it never started. What you can do in this case, you can try to rerun the job and just delete this one. It can happen from time to time, it's very rarely. But for the sake of time, I copied exactly this output file from an history I had pre-prepared, so we can continue our tutorial for today. So your output file is now called get flanks on data 2, as we had as input the genes file, which we created in the second step of today's tutorial. And you see that we have the BED format and the MM9, and to check whether what we did was really working, we can now again open the data 2, so our genes file, and the newly generated one, and compare the start positions that we have. So I open the newly generated file and also the gene file. To compare them side by side, I will make it a bit better visual for you. So on the right side, now I have the genes file, so the original file, and on the left side, I have the file where we extended the promoter region. And if I compare now for the very first line, the entries, so the original one had, sorry, I'm very bad with numbers. Okay, so 134,212,701 was the original start position. And now we want to add the promoter region. So we need to go upstream. It's on the plus trend, which is also important. So upstream, we reduce 2000s because this is the region that we added in hopefully detecting the promoter region from this. So it's very simple. This number should change into a 0, right? And voila, we have exactly at this position now a 0. So we have upstream added 2000. And now the new end position is just this position plus 12,000, which then ends up in this number. So you see that we now have shifted start and end position according to what we specified in the get flanks output. As this name again is not that meaningful, this get flanks on data 2, we can rename this file by clicking on the edit button and then give, for example, the name to the file promoter regions. And of course, we need to then save the file. And voila, we have a new file name. So I can now safely delete this run that failed for me because we have the file here. I hope it worked out fine for you. Next, we can, because now here we have as format the BED format, we can of course also change our peak region file from interval to BED. To do this, to change formats or databases or anything with a file, we again click on the pencil icon. But this time we want to go on the data types tab because we want to change the data type. And here you have two options. You can either assign a new one or convert the data type. It will, in the first place, just switch the data type here, and in the convert option, it will create you a new output file in your history. And we're going to do this now here. So we want to convert our peak region files to BD format. And this was already selected because the underlying checking already checks what makes sense to convert it to. If this is not selected here, just search for the conversion into BED. And now I can click on Create Dataset, which should now create a new entry in my history that is the converted peak regions file, which is changed then into BED format. So here now the process is running. But we already see that the format here now is BED, and the database we did not change, so it's still MM9. So the conversion for me is finished, and you see that the name is peak regions. So the same name as we had before, but as we converted this into BED, we can rename it and just say peak regions and add BED in the end, so we know what we did here. BED, so peak regions BED and save. The next step, what we can do now finally, is actually to intersect our gene file, or to be more specific, the promoter region file, with this peak region BED file to get the overlapping regions, and therefore for each of the peak regions, a name for the gene. So I will go back to the tutorial to find the intersect tool hands-on box. So we want to do now an intersection, and there's a tool called intersect. I click on the tool version, and what we want to return is overlapping intervals. That is fine. And as our first input data set, we want to select the promoter regions actually, because the order is important, because we want to keep the gene names, and all of the columns which are in this first inserted file will be kept. So then as a second one, we take our peak regions BED, that is fine. And it's also okay to search for at least one overlap. And with all of this parameter specified, I can run the tool and hopefully get an output soon. And with this output, you now have finally a list of gene names where you know those genes overlapped with the original peak region files. So you have some material detected on these genes. And this is important because you may want to research further or ask many different questions regarding those genes that were found. So from now on, a more in-depth analysis could start. But for our case and just seeing what tools to run, we are very happy with this result. One option that you could do now is, for example, to visualize the data a little bit. And one question that you would like to visualize is, for example, so for which chromosome, how many genes were detected on which chromosome? Because at the moment we only have a list with many regions, but yeah, it's hard to interpret anything, right? And therefore we can use this chromosome line and just count how often a chromosome is how many lines with chromosome 1, chromosome 2, and so on occur in this file, and use this then as a base for visualization. So let's use a tool that is called group. So in the next hands-on box, you will find the tool group. And by clicking again on the tool name, we can specify that here in column 1, we want to detect how many lines we have. So for this, we need to insert an operation, and we don't want to have the mean, but the count. For column 1, and we don't need to round to the next integer. And with this, we specified all of the parameters. So we want to group on the column 1 and perform the operation on these groups by counting the number of columns 1 within each of the groups. And therefore, I can run the tool and see what output I get. Our data is grouped now, and I can open up the output. And here you see that for column 1, the chromosomes, we have now the number of counts, how many genes fall onto the different chromosomes were detected in our peak regions file. And if you want, you can try to, for example, figure out which of the chromosomes has the highest number of genes. And in our case, it seems to be chromosome 11 with 2,164 numbers, regions detected in this chromosome. The order of the chromosomes at the moment is a bit arbitrary and not so nicely readable for us, because we would expect chromosome 1 and then not chromosome 10, but chromosome 2 in the next line. So therefore, we can use a tool that is called sort. So if you go back to the tutorial and click on the tool sort, we can actually sort this group data. So input is our last output. We want to sort on column 1, that is fine. And we want to sort in ascending order, that is also fine, but we don't want the numerical sort as it is at the moment. Like we have chromosome 1, but then chromosome 10, because 1 and 1 is in the numerical order, it comes after each other. But we want to sort it neutral, and therefore we select this ordering. And with this, I can run the tool and see if we get a nicer sorting or a nicer order within our output file. The sorting is finished, and we can have a look at the output file. So let's open the preview. And now you see we have them nicely sorted, starting with chromosome 1, chromosome 2, chromosome 3, and so on. And this sorting now is in a nice format that we can also visualize the output, so the counts. One option how to visualize such count data would be, for example, a bar plot. And within Galaxy, we have the option of producing many different plots. And you can visualize data by either clicking the Visualize button within your file, or you could also click here on Visualization. But let's do it from the file directly. And here we can select bar, line, and scatter plot. And wait until the preview comes. Yeah. So now we have a nice bar plot, but we can, of course, also change and adapt this plot a little bit. First, it would be nice to give it a meaningful name. So, for example, gene counts per chromosome. And we can also switch, for example, to the settings and change the X and the Y label. For example, I can say X to chromosomes and the Y one to maybe just counts. And I mean, you have many different options. You can also play around a little bit. You can select a different color. Maybe you want to have pink or green, whatever you like. And yeah, just see, you can also select another plot, but a bar plot in this case makes more sense. I would encourage you to just play around with the options here a little bit to familiarize yourself here. And once you are happy with the plot, you can save it by clicking on this Cloud Upload button, which will save your plot, and you get the message successfully saved. If you want to reopen the plot, you will find it if you open the visualizations in your tool panel, and here you have this blue button which says saved visualizations. If you click on this one, you see my visualizations shared with me and public ones. And in my visualizations, you should have a plot that was just saved a minute ago. And if you open up this one, you can see the plot again once it comes. Yeah, here you will have your plot again. Yeah, so this visualization is a nice option because most of the time, if you do any kind of analysis, you want to visualize the data you analyzed at the end. And I would encourage you to just see what options there are and just play around with them and see whatever fits for your data and makes sense. It might be that you have a very similar task in the future, or you want to share whatever you did with a colleague, and therefore it would make really sense to create a workflow out of your history entrances, so out of the analysis you did. And you can do this in Galaxy, which is really nice. So in the right section, so in your history section on the right side, you have history options on top. And if you click on this history options, you see the option extract workflow. If you click on this one, you get the possibility to extract a workflow. And here you can now either take all of the steps we did in this analysis, but of course, the analysis we did was very tailored to the input data we had. For example, we checked this include only the last lines to just check whether we have what kind of chromosomes entries we have at the bottom of our file. This might be not needed if you rerun the workflow several times. So you can maybe also exclude this step. And it is also not given that your input data don't have the CHR. So we can also exclude the replace text ones and also again the select last ones in the end. So I deleted this step of the flanked regions, get flanks, and imported a new one. Therefore, I have two here. You should have only one entry here. But as this is also very tailored to our analysis, we don't need to include this get flank tools per se. But of course, we want to have the convert the genomic regions. Sorry. Convert the genomic regions, of course, you also don't need, because the intersect tool, which we will include, can also take this interval format, or you have files that are already in BED format, and therefore you don't need to convert it. So to make this pipeline very general, we can exclude all of the little steps we did that made it very specific for our case. But the intersect tool, of course, this is the main operation we did today. And if you want, you can also leave the grouping and then the sorting for later on, just to see then how many genes per chromosome were detected. Okay, with this, I can now create the workflow. And I want to edit the workflow a bit. So I click on the Edit button, and this will open the workflow editor for me. And here you see that we have our steps. Sorry, I'm faster than my computer. Okay, now I can, so if you click on one of the tools, you can move it around. I'm just moving it a bit so we can see a bit better what we have as input here, or as steps here. So we have two input files, our gene files and the peak file. Then as the operational step, so the first tool is the intersect tool, where we intersect the gene file and the peak file, and then we group and sort the output. As we deleted many steps in between, there is no connection between the intersect and the input files, but we can now, by just clicking on these arrows, create this connection. So our peak file, we want to have as a second input, and the gene file we want to have as a first one. So you click on the file, this then opens up this line which you can drag until the input file again. And now you have a nice workflow already created. You can somewhere, yeah, here. You can auto layout, click on auto layout to get a nice overview of whatever you did. So this just try to make the order of your steps a bit nicer. So, and once we have this, we can click on the save button because we are happy with our changes. And with this, you created your first workflow. Voila. And we did now the first part of the Peaks to Genes tutorial. If you are interested in a more sophisticated part, then please stay just within the tutorial now. Or if you don't want the more sophisticated part, but just want to know how to share now your results, please jump to the end of the video. We will mark the time stamp in the description. As a more sophisticated approach, we could think about of doing the same overlap we did before, but this time not just overlapping this broad peak area that is specified in the file by 1, because the peaks can be very broad and there might be not that much content at the borders, but we could just search for an overlap with a peak summit. The peak summit means the area where the most amount of reads fall into. So for this, let's open up a new history because we want to perform a new analysis. I go back to my main interface by clicking on Galaxy, and I want to open a new history. I can do this by clicking on the plus, and this will open up a new history. I can rename this history by GTN tutorial peaks to genes part 2, or more sophisticated part, whatever you want to name it. And then I click on save and I have a new fresh history. As we already have our peak file as an input uploaded in the previous history, we can import this peaks file from there. So there is in the history options the option to show history side by side. Here I already have my latest history I worked on within the multiple history view. If you don't have that, just go on select histories and add this one as well. So I will search for the peak region file that we have specified, because there we already did all of the steps we excluded from our workflow analysis. So I just drag and drop this file into the new history, and voila, I have this file now inside my history. And by looking at the data a bit more closely from this output that we have, we can see that in column 5, actually the position of our peak summit is given. So from the start side, 37 base pairs plus, we have our peak summit. And to actually use this information now in a new file format, we can convert this file and generate out of this information a new start position that starts exactly at this peak summit and maybe just goes 1 base pair more until the end and computes this as the stop of our peak summit, so that we have start and end position of the summit in the file, and then we can use this file for the intersect later on. Okay, let's jump to the tutorial and see the tool it suggests us to use. to do this. So we did the visualization and the creation of a workflow, and we are in the more sophisticated approach part. We created a new history and selected the peak regions file, and now we want to create a peak summit file. And this we can do by using the tool compute on rows. So I click on the tool, and my input is the only file I have in the history, which is fine. The input has no header line, and we want to extract. In the add extraction, the start position. So our start position is here in the second column. So C2. And this we need to take plus our way until the peak summit, which we have in column C5. And maybe you see that I said integer and then in parentheses C5. I do this because a peak summit can also fall in between bases, and therefore we round down to the lower one to have an integer number here, in case that happens. And that's okay, but we also want an additional end position, so we need to insert another extraction. So this extraction will be, we have 7 lines, so an 8-line, sorry, not line, column. An eighth column will be created by the first extraction, and therefore we want to take this new summit start position, which we can find in C8, and we say here +1. And with this, we specified the 2 start and end positions we want to create for our new peak summit file, and I can run the tool. So we have now created our peak summit file. That includes now 2 additional columns, the column 8 and the column 9, with the start and end positions of the summit. So we can rename this file into peak summit file by clicking on the pencil, waiting until it opens the edit window, and renaming the file into peak summit regions. And I have to click save to also change the name. For the detection later on, we only need actually our chromosome line, which is in column 1, and then the start and the end positions. So we can use another tool that is called cut to only cut out the columns that we want to have and put them again into position 1 and 2, as will be expected by our workflow. So I click within the tutorial on the tool cut, and here I can specify that I want to cut out column 1, which is already given, but then column 8 and column 9. It should be separated by tabs, which is fine, and It should use the peak summit regions file, which is also fine, so I can run the tool. The output of the cut tool is now in tabular format, but the workflow we want to run in a little bit that we just created is expecting the interval format. Therefore, we can change the format to interval again because, as you remember, interval format expects the first 3 columns to be defined and then how many ever columns additionally, but yeah, we don't have this additional columns here, which is also fine. So you can do this by specifying, by clicking on the data type. And now we can assign a data type this time by choosing the assign section. And here we search for interval because we just want to change the data type here. And I click on save to then hopefully change the format to interval. You see it's already detected, but it's still processing and running. However, we can proceed because as a next step, we actually need also our gene names file again. And if you remember, I cheated before a little bit by telling you we want to have the gene names, but what we actually had was the RefSeg IDs. If you remember, the names were called NM and then underscore a number. So we did not actually have the name, but the IDs. If we want to use this time, maybe the names, we can import another file that is stored in the data library for us. You can either search in the data library for this, or you can import it via a link from Zenodo. So I will open the tutorial to get the link. If I scroll down a little bit, you see the next section, which is called Get Gene Name. And here our first hands-on is to the data upload. And here I can just copy the link for uploading it. And we can again upload via link. So you would also see how to do it from the data library if you expand the tip here, but we will do it again via the link. So I go back to my Galaxy interface, click on upload, and again on paste and fetch data. And here I include the link. And we can this time maybe already call it the name that is given in the tutorial. You can go back to the tutorial to get this one. And we can also specify the genome, which is the MM9. And upload the file by pressing Start. It should automatically detect the BED format. So we see if this auto detect here for the format works. But yeah, let's see. If not, we can always change it. So you could also change the name and set the database after uploading. But this time we already specified it before the upload. So I close the upload and wait until my file gets green. The upload is finished for me, and we can have a look at the file attributes. So the format is interval here, and database is mm9. And we see now, if I open the view button, that actually in column 7, we have the names of the genes given. So this is great. We can rename the file into just mm9 refseq genes, and also maybe give it again a tag genes, so we know which file we are using later on, or which was used for what. Okay. Then let's repeat our workflow. If you click on the left side on the workflow icon in the activity bar, it will open up all of the workflows you have. We should maybe have renamed the workflow that we just created before. But you can also see the time when you created this workflow, so I can be sure that this is the one we created within this tutorial. So to run this workflow, I can now go on Run Workflow, and I can click on Expand Workflow Form to get a bit of more options, because when you expand this workflow form, you not only see the input files, but also the steps that are happening later on. And as we know now, we don't want to, for the gene file, we don't want to use the column we used before for the IDs, but now we want to actually use the gene names. We need to adjust this. So first we select our gene file, which is already selected, and as peak files we use the one which was the output of cut. And I'm sorry, I think I told you something that we will do in a second, but this time we will not change the gene names, but the grouping. So let's quickly open up the tutorial just to be safe in what we should do as a next step. So I can also directly jump to the more sophisticated part. And we are in the section where we want to repeat the workflow. We already opened the menu, clicked on Run. Then we choose the input file first, the genes, and then the peak one. And then we can run the workflow. This is the first hands-on. So I'm sorry, in this step, we will not change anything. We will just do it as we did it before and generate the same output, but with the new files that we created. So I will pause the video until the workflow is fully run. You can see the process here now, which tool is run, and step by step, all of them will turn green. So my workflow is almost done. I'm just waiting for the sorting job. But as this just sorts the output of the group, we can already see if or what kind of output we have now. So you see that the numbers changed a bit because we have a different genes input file, but as in our first analysis, we get the counts of the peak summits that fall into the different chromosome regions. And you can see with the this different input gene file and only accounting for the gene summits, the numbers have changed from our original file, and we of course get less because we are more specific this time in which one we are counting and which one not. So sorry for the confusion before. You could of course repeat this analysis by counting how many summits fall on the different genes, if there are genes that have more than one summit. And for this, we can also either just rerun the grouping because we have in our intersect file, we have all of the information we basically need to perform this. We have the in the column 7, we have our names. And for example, you see already here, we have 3 summits on this gene. So you could either just rerun the tool here, or we can, just for the sake of repetition, we can rerun the workflow again. But this time we will actually change one of the parameters in this group tool. So I click again on the workflow or in the activity bar and select our generated workflow, click on run, and again I expand the workflow options because this time I actually want to change something. So I select again our gene file and our peak file, so the cut one, the output of the cut tool. And then within our group tool run, I have to change the parameters in the count section. So we want to group, of course, by the gene names. So first I have to change the group on column 7. And I also want to count on column 7. Oops, sorry, I was too fast. So not 77, but only 7. And of course now, so if you remember, our sort tool is just sorting chromosome 1, chromosome 2, and so on in an order that makes sense for us. But this time, it doesn't make sense to sort the gene names. but we want to sort the actual counts to see what gene has the highest number of counts. So we also need to change the sort tool to sort now on column 2, because this is the count numbers. So you can see it here. We want to change these numbers this time. So on column 2, we want to sort in descending order. And the sorting type should be numeric. So we also want to change the flavor to fast numerical, which is fine. Okay, so with this changes, we changed the group column and also the column where they should be counted on. So the group column, the column which is the groups that should be counted. And then in the sort one, we changed the column that should be sorted is now column 2. And the order is descending, and fast numerical search is fine because now we have numbers. Okay, and with this, I can rerun the workflow again. And you see that it again opens up the process of our workflow. So of course, we already have the inputs. And now step by step, our insect group and sort tool will hopefully become green. And with this, we are almost at the end of the tutorial. You can have a look at the group or the sorted output file once they are finished. But I want to show you the last section of the tutorial, which is share your data. So you might want to make your data publicly available for everyone. You want to share it maybe with a specific user using an email address, or you want to provide a link to someone that can then use that link to import your history. And this is possible if you click again on the history options. Then you have the option share and manage access. And once you click on this, you can drag the pointer in make your history accessible to on. And now you already made your history accessible via a link. You don't have to do this. You can always retract it. So now it's again not accessible via the link. But you can also, if you have a colleague, a friend, whoever, and you know this one is also a user on your Galaxy instance, you can provide the user identification, so the email address or the user ID, and share it with this user by just entering the user's email address here and then click on save. I don't have this at the moment to show you, but there is a third option I want to show you. So when you again make the history accessible, it's again now accessible via link. Here you can change the URL if you want to, but most of the time there is no need, and you can directly copy it. But you can also make it publicly available for everyone. So if you also check this bar, your history will be publicly available. And if I click now on the publicly available histories. It should open up all publicly available histories, and you see that I just shared this history with everyone who is using usegalaxy.eu. And we can have, yeah, and with this, we are at the end of our tutorial. Thanks a lot for following along. I hope you are a bit more equipped now to handle Galaxy, and you are excited to start your first analysis or continue with another tutorial. So all the best, and see you maybe in another video. Bye.